Hello everyone, welcome to MES e-learning channel. So now we are going to have the last lecture for the genome editing series of lectures. So the most revolutionary genome editing tool, that is the CRISPR Cas system, that we have already learned in the previous lecture, that how it is used in the nature. In this lecture we will see how it can be exactly used in the cells for the modification of the DNA. And that definitely opens up a lot of possibilities in research, medicine, environment, and beyond. So, as we said, that I had already said that in case of the nature, what happens is the Cas9 protein has to get connected, has to get combined together with the CRISPR RNA, which acts as the guide RNA for this Cas9 protein to create the double-stranded break. So once this guide RNA recognizes and binds to your DNA sequence, it will activate the Cas enzyme, which will cut the DNA and then bring about or will create the double-stranded breaks, and then followed by the cellular repair. So cellular repair either by NHEJ or it would be your HDR, depending upon what is the need of the genome modification. So this is the structure of Cas9, where we can see that there are different domains. So you have Rec1, you have Rec2, you have RUVC, and each of this domain is going to have different different functions, different roles. So you have the PAM interaction section, which is going to identifying the PAM regions in the sequences, so that it will identify that this is the sequence that we will be able to cut, and followed by, so that is the reason why you have a very important structure of the CRISPR RNA. The CRISPR RNA structure is an hairpin loop kind of structure. So in the nature, you have a two-piece RNA. So you can see that you have the guide RNA region and you have the tracer DNA region. The tracer DNA region along with the guide RNA, it will combine together to this particular Cas9 protein. But when we are doing the genome editing, the researchers have combined together both this guide RNA and your tracer RNA into a single guide RNA, which is called as your sgRNA. So it is for the simplification purpose. So let us now see the comparison between the natural CRISPR-Cas system and the engineered CRISPR-Cas system. So in the natural process, which we had already discussed in the earlier lecture, we know that you have the CRISPR locus in which the genome of the viruses gets incorporated, and that will get transcribed and then make up this entire guide RNA and the tracer RNA together, which goes and activates the Cas9 protein. So here when you have your engineered CRISPR-Cas system, so this entire there is a plasmid which is going to be designed in such a way which will have the genes for your Cas9. Along with that, it will have the entire genes that will help in expression of a single guide RNA. So when both these are expressed, so these are your expression plasmids which are going to carry the promoter sequences. So once it gets expressed together, this guide RNA and your this Cas9 protein will come together, activating the Cas9 protein and directing it towards the target molecule such that it will create the double-stranded breaks. Okay, so the double-stranded breaks are going to be created because of this activated Cas9 activity. So this is the general protocol. I have given an example of how this CRISPR system, CRISPR-Cas9 system can be utilized or can be helpful in editing a particular genome in plant cells. Okay, so let us see this. So first step that we have to do is select the genomic target. So along with, like, there is a 20 base pair sequence which is going to be selected, which needs to be modified. along with a PAM region. So this PAM region will be your NGG. So N means any of the nucleotide can be there, and it will, so we will be using different types of online tools, that is your bioinformatic tools, to check whether this 20 base pair sequence is common or not. It has to be unique so that we can reduce the off targeting. Off targeting is not taking place, we have to take care of that. Once we have selected this genome target, then we will go for designing the sgRNA. So this is going to be expressed using small RNA promoters. So like you have your U6P or U3P, these promoters are going to be combined together with this particular sequence. So the first nucleotide in the guide sequence is always the G. So if it is U6P is used, and it is A if your U3P is used. So depending upon which promoter you are using, there could be a slight changes that could have been applicable. So you can see in the diagram also that how your gene is taken, the target sequence which is going to be connected to your PAM, and then you are designing the whatever the plasmid is there. So you have the guide sequence, guide RNA sequence, followed by your SG sequence backbone and connected to the promoter sequence. Okay, so when it gets, so once the assembly and everything is done, the construct is going to be made. So in the same way, the plasmid has to be converted. Okay, and in the assembly we have to also incorporate the Cas9 gene, so that once it's expressed, both the things are expressed together. Then once the construct is ready, it has to be delivered to the plants. So this delivery would be done by different ways. You can use protoplast transformation, agrobacterium transformation, or callus bombardment. So different ways of delivery can be utilized so that you can this whatever the assembly of your CRISPR-Cas9 system is there, it is delivered inside the cell. Once that has been done, then you have to regenerate it into the plant and also check whether the transgenesis has occurred or not. So you can have different types of your selector proteins also incorporated in the assembly, so that it will help you in screening the mutants. Or you can just go for next generation sequencing to check whether the mutation that you needed or the modification that you needed in the genome has been incorporated or not. So this is how your entire process of your CRISPR-Cas9 can happen. So again, the ethical considerations are also there. So whether we have to use all these things responsibly, definitely careful guidelines have to be followed. Germline editing is still a concern, but here I would like to tell you that a few years back there was an incident when there was some Chinese researcher who had developed some twin girls where the HIV genes were altered. HIV genes were altered means the target for the HIV, those targets were altered so that they would create the resistance towards this particular virus. But it still remains an ethical issue that whether such kind of your editing in human embryos should be allowed or not. Because all these are going to go for equitable access is always going to be a concern, because all these tools are going to be expensive, and the access of such kind of your tools to all the people is going to be a concern. So as the technology advances, definitely we would be able to bring down the cost of it and use it as a global health requirement and develop into such a way that it could be used in equitable purposes. So public awareness is also very much crucial. There is always thinking whether such kind of your genetic modifications we should go for it or we shouldn't go for it. But this is a little, such kind of your genome modifications are a little bit different as compared to your transgenic modifications, because in transgenic systems we are incorporating some other genes inside the like for example, if you are taking a plant genome and then editing it into a human genome or any kind of your animal genome. So such kind of combinations are being done in case of transgenics. But when we are talking about genetic modifications, so the indigenous genes itself is modified. So a lot of debate is going on whether it should be considered as a transgenic or whether we should accept it for different applications which are there. So advances, again, improved specificity. So a lot of research is still going on that how we can make this particular system more and more precise as well as accurate, and novel Cas9 enzymes, like apart from Cas9, what are the other Cas enzymes which can be utilized, which will have a better functioning. And again, delivery optimization, which has already been a problem in all these systems for genome editing, that also always a problem, and this is being overcome by a lot of research, like what efficient ways we can come up to. So future, definitely CRISPR Cas system is revolutionizing the entire system, where we can modify the genes and traits in plants, animals, humans in vivo, and personalized medicines, like we can go for the targeted therapies and tailored treatments for genetic disorders and diseases, sustainable agriculture production, so that improving the crop yield and developing resistance for the your diseases, as well as for say drought resistance or any other kind of your changes that you want to do, and improve the nutritional quality that can be done, and disease prevention, as I have already given an example how for HIV is being done. So similarly, there are some genetic predispositions for other type of disorders also, so that can be eliminated, so that the people, the human beings, can become resistant to such kind of your diseases. So these are some few possibilities that are there for every genome editing tool. So with this, I'll stop here for everyone, and I hope I could explain all the methods of genome editing tools. Thank you so much.